Okay, so hi everyone, welcome everyone. I'm Massimiliano Tamburrino. I'm one of the um, organizer of the former One World ABC. We went through 5 seasons with the One World ABC, starting in April 2020 as a way of gathering people during the pandemic. Um, we initially were, I think, even bi-weekly seminar, and then become a month. And then in the last year, we tried to widen the scope and the goal of the seminar, switching more broadly to approximate Bayesian inference, but somehow this wasn't really fully perceived. So that's why we decided to come out with a new name and new seminar in scope to some extent. So that's why we then switched to the one word ABI, so the one word approximate Bayesian inference. We have already 3 great speakers lining up, and also some new member of the organizing committee. And so I would like to welcome Stefan Radev as one of the new member, and Stefan will host this talk today.
All right, should I take over?
Yes, please.
Can everyone? Okay. All right, so I'll be very brief. Thank you for accepting our invite. Uli, a few words about you. Ullrich Köthe is a very special guest to me. So not only is he a role model researcher, but he also wholeheartedly adopted me during my PhD in the idea space of deep learning for science. He also advised me during my postdoc at Heidelberg University. He's among the key originators of our Baseflow project and certainly one of the main reasons why I'm here today. So it would certainly be an understatement to say that you are an extremely versatile researcher. You've been working on computer vision for more than 200 years now, or at least that is how far back his knowledge of all possible algorithms extends. He and his team have pioneered many techniques and deep learning architectures. Some of them you know, conditional invertible networks, incompressible flows, generative classifiers, and more recently free-form flows, of which we will hear a lot more today. And most notably for this seminar, Ullrich and his team were the first to systematically analyze challenging inverse problems with invertible networks and provide convincing empirical and theoretical results suggesting that we should take neural networks for Bayesian inference seriously. Okay, his team has also provided what I believe to be the most well-founded theoretical analysis for understanding the universal density estimation properties of normalizing flows, and have recently developed a really cool method to turn any neural network into a normalizing flow. So Uli, we're very glad to have you here for our Kickstart meeting. The stage is yours.
Okay, thanks for the kind introduction. And actually, many of the results I'm going to present today are actually Stefan's results, which I was just happy enough to be the supervisor of. My group is called the Explainable Machine Learning Group. And so we are interested in interpretable models. And one way of achieving this is to combine deep learning with physics prior knowledge. And I will show a few ways of how you can do this today. And freeform flows are just the newest incarnation of the general idea. So before I go into these things, I would just like to present a categorization of the deep learning generative model zoo, because it has become quite confusing recently with many competitors. So what are we talking about? The goal is to learn some complex probability density P of X, or some also complex conditional density P of X given Y, when we only have training set, so a bunch of X values drawn from some true distribution, or a bunch of pairs of X and Y's also drawn from the true conditional distribution that we do not know. And I mentioned complex here because neural networks especially shine if these distributions are complicated, so that the classical algorithm no longer work very well. So, and what can we do with a model once we have trained it? So we can use it to sample synthetic data that should hopefully be indistinguishable from true data. Yeah, then we can also calculate the probabilities, and more importantly, can compare the probabilities of different data points, so to judge which is more plausible than the other, or also we can compare the probability according to competing models to judge which model is better. And these two basic fundamental things, sampling and calculating probabilities, can then be used in a ton of downstream tasks like Bayesian inference or solving inverse problems and so on. So as I said, the zoo has become quite complicated. So the word map shows some of the more famous algorithms. And the first question is, which of these should you pick and why? And so first of all, if you look deeper into how these models work, you will notice that most of them follow the principle that they use a simpler distribution and then create the complex distribution by some transformation. So this is how it looks like. So we have the axes that are drawn from the model distribution. And what you actually do is you draw z's from some simple distribution, such as a standard normal, and then transform the z's into the desired axis. This is called the decoder, the function g. And if you pick the right function g, then the distribution of x will actually follow your desired target distribution from reality. So this is the basic principle that most of these algorithms use. So, and now they distinguish how they are different by how the function g is implemented. And I found that one very interesting way of categorizing methods is by exactly how they express the probability p of x in terms of the decoder function g. And this is mathematically called the change of variables formula, because we change the variables from x to z. And that's why I should start with explaining what a change of variable formula is. So, and normalizing flows are maybe the most straightforward realization of this idea. And that's why I start with normalizing flows, which are also the workhorse model so far in our research and in Stefan's research, I think, as well. So here is how it looks like. So we have an encoder-decoder architecture. So we have x's, which are encoded into latent representations z, also called the codes in this context. And the encoder function is called f. And then the z's can be decoded back into x's by the decoder function g. And in case of a normalizing flow, the dimension of z is the same as the dimension of x. And the decoder function g is bijective, and f is just its inverse. And in the standard implementation, the coupling layer normalizing flow, encoder and decoder are actually not two different networks, but the same network running forward and running backward, so-called invertible neural network. So, and if you use this architecture, then the change of variables formula looks like this. So we want to calculate the probability of some generated sample, g of z. This is a synthetic data point. And it can be calculated as the raw probability of the sample z in latent space times a correction, which is the inverse of the Jacobian determinant of the decoder function. Yeah, and this term adjusts for the volume change, the local volume change around the point Z as you apply the function g. And this is essentially the same change of variables formula that you are familiar with from integration, yeah, in calculus. So, and this has been known for a long time. And in 1D, it's the basic of how distribution or how samplers are implemented in software libraries. So here, usually you start with a uniform distribution over sets in 0, 1. And then the decoder for the simple distributions, so like the Gaussian, are usually known in closed form. And for the Gaussian itself, it's just the inverse arrow function. Yeah? So the decoder is a scaled version of the inverse arrow function. Yeah, and the principle of normalizing flows is to generalize this basic idea to arbitrary dimensions and arbitrarily complicated distributions. So here is an intuitive illustration of what this means. So in this case, we have chosen the latent distribution Z to be the standard normal, so a L-shaped curve. And the distribution we would like to generate is a mixture of two Gaussians. Yeah, so this is already a bit more complicated than a single Gaussian. And intuitively, you imagine that your probability mass in latent space is a pile of sand. And you have the task, so the decoder has the task to move this sand around until it has the shape of the green target distribution. Yeah, so here this is easy. So what you do is you split your sand into two halves and then move the two halves to the right locations and then shape the distributions according to the target distribution. Yeah? This is what the decoder is supposed to do. And here are the terms again. So we have the latent distribution Q of Z, we have the target distribution P of X, and we have a transport map that acts between them. So F, the encoder, and G, its inverse, is the decoder. And then mathematically, this is often written as what's called the push forward. Yeah, this is this hash symbol. Yeah, and the decoder pushes forward samples from Z space to X space. And I already showed the change of variables formula. So now, how can you implement this in practice? First, you notice that training is very easy, at least conceptually. So we would like to maximize the probability of our training set under our model. And this is, of course, equivalent to minimizing the negative log likelihood of the training data under the model. Yeah, and then if you insert P of X according to the change of variables formula, you end up with a pretty simple expression. So you have the negative log likelihood of the latent codes times the logarithm of the determinant term, yeah, the volume change correction. Yeah, and this is what you later use as a loss function, because it turns out that it's nicely differentiable and everything. So the problem is, of course, that you need the logarithm of the determinant of the Jacobian. And this used to be intractable, but there are a few tricks, and some of you I will show you today. So the simplest trick is that when the Jacobian is a triangular matrix, then its determinant is just the product of the diagonal elements. Yeah, and this is the basis of what's called triangular transports or Knote-Rosenblatt mappings, where you use a triangular decoder function G. Yeah, and in the standard implementation of normalizing flows, we exploit this principle layer-wise. So the entire network is not triangular, but every layer is triangular. And then we have that the determinant of the entire network is the product of the determinants of the layers. Oh, here's a typo. Should be, I know it's correct. And then the determinant of each layer is the product of the diagonal elements, and the determinant of the entire network is the product of the individual determinants of the layer. So here's the typo. Here is a determinant missing. So, and this can be realized in the easiest case with what's called the coupling layer architecture. And this is how the coupling layer architecture looks like. So, and this especially is the affine coupling layers, which is the most common implementation. So, and the coupling layer splits its input, here called X, into two halves. So we have X has D elements, and X1 has the first D over 2 elements of X, and X2 is the subvector with the second half of the elements of X. Yeah, and then the output of the layer has the same split and is then concatenated into Z, the final result of the layer. And then we see that the lower half of the vector X is just connected by a skip connection, so it does not change at all. Whereas the top half, first D over 2 elements of X are transformed, and they are transformed affinely. That means they are multiplied with some scaling factor and shifted by some offset. Yeah, so this is the forward computation. Z1, the upper half, is X1, the upper half of X, times scaling factors plus offsets. And the lower half is just a skip connection. And the trick is now that the coefficients S and T are now functions of the lower half. And this makes the whole thing very powerful. Yeah, so here's an illustration. So we split, we calculate the scaling and offset coefficients of the upper half using the lower half of the vector, and then do the calculations and concatenate the results. So the upper half of the affine transformation and the lower half from the skip. So, and since this is a affine function, its inverse is easy to calculate. Yeah, it's just Z minus offset divided by the scaling factors, and then the network runs backwards. So we split up the Z into the upper half and the lower half, and then we use the lower half to calculate the coefficients, and then we calculate the affine transformation and concatenate the two results. Yeah, and the crucial insight of this architecture is that the networks S and T, which calculate the coefficients, are always executed in forward direction, regardless whether the entire layer is executed forward or backward. Yeah, so the blue networks S and T do not have to be invertible, and still we can make the entire coupling layer invertible. Yeah, and in practice, S and T can be arbitrarily complicated neural networks depending on the application. So, and one thing that I do not have time to mention, of course, if you stack many of these coupling layers into a deep network, then after each coupling layer, you will have a permutation or a rotation of your variables so that it's not always the same elements of X that end up in the skip connection. Yeah, so every element of X should be transformed at least once. And in practice, it's sufficient to just choose random rotation matrix in order to make sure that this works. So this is a very brief introduction into how a change of variables formula helps us in learning a generative model. And now for an application, which is, of course, the application that was the basis of Stefan's work, simulation-based inference. So the idea is that in many fields of study, we already have prior knowledge in the form of a simulation. And as you remember, the point of the talk was to exploit prior physical knowledge about your problem in neural network training or architectures. So, and the existing simulation allows us to train the neural network purely from synthetic data generated by the simulation. And this is often very good because real data tend to be scarce and expensive, and they should not be wasted for training, if at all possible. Yeah, and once the network has converged using the synthetic data, we can then apply it on the real data in order to do inference. Yeah, so here is, so the gray box is the training, and then the outside stuff is the real observation. And we are interested in inferring some hidden parameters x from some observable quantities y in this case. That is always in Bayesian inference. And we have seen very good results in many different and very diverse applications. So from image analysis over physics, cognitive science, to mechanical engineering and medicine. And I will show today the example from COVID modeling. So here is a slightly extended SIR compartment model with, in this case, 6 compartments. So we have the susceptible people who are still healthy but may get infected, the exposed, which are still symptom-free and cannot spread the disease. So this models the incubation period. Then the carriers, which are not yet diagnosed of the disease, but can already spread it. So these are the dangerous people. And then we have the infected compartment where you know that they are ill and you can isolate them in order to reduce the potential to spread the disease. And then people can either die or they can recover from the disease. And here are the most relevant parameters of this model. So 1 over gamma is the incubation period, so the time you stay in the exposed compartment. Then we have the infectious period, which is the period to detect the infection plus the period to recover from it. And we have the fraction of undetected infections, alpha. This is, of course, very important for health planning and countermeasures. And we have the infection rate, which is a function of time, because it depends on countermeasures, and it also depends on how the virus mutates. So here is the same thing written as differential equations. So, and then to enhance realism, you also need to model the observation error. So for example, in Germany, we have this weekly oscillation, because on the weekends, our Gesundheitsamt does not report any measurements. So it always looks as if the disease was weaker on weekends. So, and then also you need to have a intervention model. And in total, this makes up for 34 parameters. And we chose uninformative or very wide priors for them in the absence of any medical evidence to the contrary. So, and the forward simulation is easy. You just take the differential equation and here the observation model and the intervention model, and then you do 50,000 synthetic disease simulations. And these are the training data, and then the network hopefully converges to something useful. And then, so here's the summary of everything. So here we have the time series coming from the simulation. So 3 series of observations, the number of infected people, so who have a positive test or had to see a doctor, and the number of recovered cases, and the number of people who died on every specific day in the simulation. Yeah, and this is used to train the network. This is the orange box, and then the blue box is the application to real data. And in Germany, we used the observations of 80 days, so from March to end of May or so, of these 3 observations. So infected, recovered, and dead people. Yeah, and here are the posterior distributions of the 34 parameters. So first of all, it works, and in the background you see in gray the prior distribution, and in the foreground in red the posterior. So, and if the posterior differs from the prior, this means we have learned something from the data that we observed. And if the posterior and the prior are the same, then the data were not informative about this particular parameter. And I will pick here the fraction of undetected infections. So, but before I show you this is the resimulation from our model with error bars of the simulation. So we sampled parameters from the posterior and run the simulation again. And if the model is any good, the true observations, which are the black dots, should be in the confidence interval of the re-simulations, which in this case is very much the case. Yeah? So that means our model actually is predictive for reality. So, and here is the promised fraction of undetected infections. So we used a uniform prior, because we have no idea what the fraction might be. Yeah, so this is the gray stuff. And then the posterior, after seeing the data, is still quite wide. So the model cannot exclude any particular value for the fraction of undetected, but the posterior also has a very distinct peak at about 80%. And this is interesting because later on, people did what's called seroprevalence studies. So they took the blood of a sample of people in, for example, in Munich, and measured in their blood whether they could find antibodies against the disease. And then they asked the people who had antibodies whether they knew about their infection. And it turned out that in the general population, 75% of the seropositive people, and in hotspot even 80% of the seropositive people, had no idea that they had COVID. So this confirms that the network's opinion that 80% of the cases went undetected might be pretty close to the truth. Yeah, I think this is the most interesting result of this study. Okay, so now enough about this example. Let's go back into the zoo and to the categorization. So the first question that arises from this look at change of variables formula is whether the model is bijective or injective, which means, so a bijective model implements lossless encoding, like the normalizing flow that I just showed. So the code Z is completely informative about the data X. Or in the contrary, the flow may be injective. So then the code has a smaller dimension, like in an autoencoder, a bottleneck model, and then we get lossy encoding. And here is a diagram of the bijective model that you have already seen. So we have the X's encoded into Z's and then decoded into X hats. And the dimension of Z is the same as the dimension of X. And then the reconstruction is perfect. And this results in the change of variables formula that we already discussed. So, and then the picture for the injective models looks exactly the same somehow. Bit of the content. Oh, yeah, okay. Yeah, so the diagram is the same, but now the dimension of Z is smaller than the dimension of X. So we have a bottleneck. And this means that in general, we cannot perfectly reconstruct our samples X. And then the change of variables formula must be adjusted. We now have the determinant of the square of the Jacobian of the decoder. Yeah, because the Jacobian of the decoder is no longer a square matrix, so it does not have a determinant. And we can recover by using Jacobian transpose times Jacobian. And to adjust for the square, we now take the square root of the determinant in the end. But otherwise, the change of variables formula remains the same. But one needs to keep in mind that this formula does not apply to the original X's, but it applies to the reconstructions. Yeah, because this formula can only calculate the probability of the reconstructed data X hat. So this is the first categorization. The second question that is useful is the model deterministic? So do we get a unique correspondence between codes and samples, or is it stochastic? Can we have several possible outcomes for a single code? Yeah, and as we have seen, deterministic is the autoencoder, and stochastic would be the variational autoencoder. And again, comparison. So on the left, we have the diagram of the autoencoder that we just discussed. And again, the diagram of the variational autoencoder looks almost the same, but now the codes are no longer a deterministic function of the data, but a conditional probability. So several codes are possible for a fixed data point. And likewise, the decoder also is no longer a function G, but a conditional probability. So we can have multiple outcomes for the same code. Yeah, and the standard variational autoencoder, both of these are realized by Gaussian distributions. So, and then the change of variables formula looks like this. We again have the term in Z space times a volume correction. And the volume correction is now the ratio between the two conditional distributions. Yeah, and this must hold for all possible set that can generate the given point X. And this is unfortunately often violated in practice. So people have not noticed that this would be a very interesting diagnostic. And we have actually exploited this in a joint paper with Stefan and Marvin and others. Okay, so this was question 2. Question 3 is the model executed in discrete or in continuous time steps? So in discrete time steps means we have a multi-layer network, a deep network, standard network, and continuous means we have a diffusion model where here again, we have the multi-layer network. So the encoder function, the decoder function is realized by a concatenation of layer functions. And then we get the change of variables formula where the Jacobian determinant is the product of the determinants of the individual layers. And in diffusion, the whole thing is implemented as a differential equation. So we initialize the differential equation with the sample point Z at time 1. So we arbitrarily set time 1 for the Z space. And then we run these differential equations and up to time 0. And no, the other way around. Time 0 is Z and time 1 is X. Yeah, so we start with Z at time 0, run the differential equation, and at time t, we read out the X. And then the change of variables formula looks like this. So the volume change term is now the exponent of the integration of all the infinitesimal volume changes at every time point, which are expressed by the trace of the Jacobian of the vector field. Yeah, so this is more expensive, but in practice, it gives very, very good samples. So it's called the instantaneous change of variable formula. And if you would like to read more about this, you can check out a paper that I wrote about these categorizations. So now enough for the general stuff. Let's move on to the freeform flows. So first, the normalizing flow, as I explained, is very good, but it has two potential disadvantages. So the coupling architecture may limit the network's expressivity. So in theory, it's universal, but in practice, you may not be able to actually make it universal by adding more layers also. And the other thing is that the coupling architecture makes it difficult to incorporate prior knowledge into your network architecture. So on the other hand, we have the diffusion models that currently generate the best synthetic data, but they are pretty expensive because you have to solve the differential equation. And they are also not interpretable. So they create data from pure noise. And the different axes of the noise in Z space do not have a semantic interpretation. It's just noise. Yeah. And for interpretable models, you would like the latent space to be interpretable. So, and the question is, can we have the best of both worlds? And that's it. The answer is freeform flows. So now we still need to calculate the change of variables formula, so the determinant, but we can no longer use this trick with the triangular Jacobian. But it turns out that modern libraries like PyTorch and TensorFlow have a function that's equivalent to this, a Jacobian vector product. Yeah, and the arguments are the function where you would like to calculate the Jacobian from, the point where the Jacobian should be evaluated, and the vector that should be multiplied with the Jacobian. And the trick is, if you use the unit vectors in the Jacobian vector product, so k runs from 1 to the dimension, then the output is just the Jacobian determinant. Yeah, so as if you would multiply the Jacobian with the unit matrix. Yeah, and it turned out that this implementation in the modern autodiff is quite fast, so that you can do 1,000 of these evaluations in 1,000 dimensions in about 10 seconds or so. But 10 seconds for 1,000 evaluations is still too slow for training, and therefore we came up with another trick, which I think, I'm not sure whether I have time to explain it. So what we noticed is that to train, we do not actually need the change of variables formula, but we need the derivative of the change of variables formula with respect to the network parameters. So that's the backpropagation algorithm. Yeah, and it turns out that even if the change of variables formula is expensive, the derivative with respect to network parameters can be calculated much more cheaply. And here's the trick. So first is we replace the logarithm, the logarithm of the determinant by a trace over a product of the inverse and the gradient. So this is just a basic identity from linear algebra, holds for any matrix. And then we replace the trace by what's called the Hutchinson trace estimator, which is essentially an expectation of matrix vector multiplications with some vector v, which is sampled from a distribution with unit covariance. Yeah, and this is easy to show by the rules of expectations and trace that these two expressions are equivalent. And the cool thing about this is that now we can replace the expectation with an empirical average. And in practice, it turns out that a single sample is sufficient to get to get useful averages for training. Yeah, and this makes the whole thing fast. And then the third trick is that we still need the inverse matrix here in the expression, which may be expensive, but when we have an encoder-decoder architecture, the inverse of the encoder of the decoder is actually the encoder, and the encoder function is readily available from the encoder network. And then we replace the inverse with the encoder, and this is the final loss term that we use. So it's really simple. So this is the same as in the standard normalizing flow, and the Jacobian term has been replaced by two evaluations of the vector Jacobian or Jacobian vector product with our networks. Yeah, so really easy and efficient. So here is how it looks like. So we have the negative log likelihood loss that we have here. And when encoder and decoder are separate networks, so remember, we want to give up the coupling layer architecture, the invertible architecture. So they are now separate networks. Then we also need to make sure that they are inverses of each other. And this can be done by just minimizing the usual reconstruction error, the Euclidean difference between the input and the reconstruction that is also used for normal autoencoder training. Yeah, and so the total loss is the negative log likelihood loss that I explained on the previous slide, and the reconstruction loss. Okay, so what can we do with these things? And here are two examples. So in chemistry, you often have knowledge about the target distribution in form of a Boltzmann distribution. So the probability of a molecule conformation X is the exponent of minus the energy divided by the temperature. Yeah, this is the Boltzmann or Gibbs distribution. And this distribution has been successfully addressed with what's called SE3 equivariant graph neural networks by the famous Amsterdam people. And what does this mean? The energy of a molecule does not, the inner energy does not depend on the location of the molecule and the rotation of the molecule. So the energy should be invariant to translation and rotation. And that's what the mathematical is SE3 invariance, and this is realized by an equivariant neural network. So, and we took this network from the Amsterdam people and just retrained it using our freeform flow algorithm. Yeah, and we now have a normalizing flow because we used the normalizing flow training algorithm, but with the physical prior knowledge of the translation and rotation invariance already built in. And on the right-hand side, you see a few samples that we generated with this generative model. And here are three benchmarks. So 3 benchmark data sets. And the first 3 rows are the results of the inventors of the network with their training algorithms. And the last is the same network trained with the freeform flow algorithm. And you see that in the second and third benchmarks, we get better results than them. Yeah, so that's why we call our paper Freeform Flows, a way to turn any network into a normalizing flow. And so you can work with the network that you have already optimized, so you do not have to throw it away and still can train a generative model. So here's another application. So the next incarnation of it, manifold freeform flows, is applicable if you already know from your prior knowledge that the data can only lie on a manifold. So in the simplest example is on the left. So the manifold is the surface of the Earth. Yeah, and so earthquakes only occur on the surface of the Earth, at least in terms of what earthquakes mean for people and destruction of infrastructure and buildings and so on, or volcanoes are on the surface of the Earth, floods are on the surface, and forest fires. Yeah, and these are the distributions that we learned with models that are restricted to the surface of a manifold. And here are other manifolds. We have already seen the sphere, then the torus, that's useful for angles, the hyperbolic disk, useful for hierarchies, and then here the Sanford bunny, which is topologically equivalent to the sphere, but has a totally different geometry. Yeah? And then the blue and yellow stuff is a distribution on the surface of the bunny. So this illustrates that it is quite easy to adapt the manifold freeform flows to various different kinds of manifolds, whereas in the traditional way of doing this, you had to design a new network whenever you would like to address another manifold. And this was a lot of work, whereas now it's pretty easy. The only thing that you need is an embedding of the manifold in Euclidean space and projection of the Euclidean space onto the manifold. So, and then the change of variables formula is almost the same as before, only that we have two additional matrices which represent a basis of the tangent space of the manifold at the points of interest. Yeah, so we need to project the Jacobian into the tangent space of the manifold before we can calculate these probabilities. Okay, and the gradient trick still works as before. The only difference is that now this test vector V for the trace estimator needs to be in the tangent space and not in Euclidean space. So, and I tell you this in order to illustrate that it's pretty straightforward to implement these manifold freeform flows if you have some basic knowledge about how your manifold looks like and how the embedding works. Yeah, and here is how the training, or what the training actually achieves. So here the manifold is a circle. So if you can imagine in the 3 dimensions, the Earth would be a surface, a sphere surface. And the latent distribution P of Z in this case is the uniform distribution. And you can imagine the uniform distribution on Earth that we covered the entire surface of the Earth uniformly with Z. Yeah, and then what the decoder learns is to shoot the sand into outer space or also into the interior of the Earth. Yeah, so this would be the yellow curve with the black arrows where the sand is transported to by the decoder. And then the sand rains down orthogonally onto the manifold back. Yeah, and if you shoot the sand in the proper way, then after raining, you will get the desired hills of your sand. Yeah, that's the intuitive principle of the manifold freeform flow algorithm. And this actually works quite nicely. So here's my last slide. In a molecule, in this case, a protein, the two angles are of particular interest, namely the dihedral angles psi and phi next to the backbone group of an amino acid. Yeah, and so here we learned, so, and these angles, of course, live on a torus, so they go from 0 to 2 pi and are periodic. And here is the torus plotted as a square with periodic boundary conditions. Yeah, and you see that we learned quite different distributions for different types of proteins. And the black dots are the ground truth data. And you see that they nicely concentrate at the peaks of our learned distribution. Okay, so this should show you the potential. So the freeform flows are a very interesting alternative to the established flows because they can incorporate prior knowledge and they support bottleneck architectures, which this I didn't show in the talk. But traditional normalizing flows cannot support bottleneck architecture. And so we are currently investigating the applications and the open problems. And if you are interested, just contact me, or you can also first read our new papers. So we have written 3 papers about freeform flows in the last year. And also, please check out the Baseflow framework, which nicely implements all these simulation-based inference thingies that I have shown. Thank you very much.